안녕하세요. 저는 한승민 박사라고 합니다. 먼저 세미나에 참석해 주신 여러분들께 감사의 말씀드립니다. 시작하기 전에 간단히 말씀을 드리면요. 발표는 영어로 진행하고 질문은 한국어로 받도록 하겠습니다. 발표를 시작하겠습니다. Today, I'm going to explain my study to identify and characterize isthmus stem cells of corpus gland in adult mouse gastric epithelium. Gastric epithelium is one of the tissue continuously exposed to harsh conditions such as mechanical stress and gastric exit. This means that it requires high regenerative capacity of stem cells to maintain homeostasis. Otherwise, it can lead to gastritis, gastric ulcer, and gastric cancer. Despite its importance and long interest in them, our understanding on gastric stem cell is limited and partial. Corpus is the main body of the stomach. Corpus epithelium consists of many glands with the structure like this. A few years ago, two groups independently identified pepsinogen-secreting chip cells marked by TROI and LGR5 as a novel stem cell population in the gland base. The chip cells are largely quiescent in homeostasis. However, interestingly, they become rapidly proliferative upon tissue injury and replenish the whole gland. However, given corpus epithelium is renewed very fast in homeostasis, once in less than a week, this means that chip cells are not a main workhorse maintaining tissue homeostasis, and there must be another stem cell population that can actively contribute to homeostatic turnover. In fact, it has been speculated there might be another stem cell population in the isthmus region because of high proliferation activity observed in that regions. But this conjecture was not proven in a rigorous way for past a few decades. With the advent of lineage tracing technique, it has become standard method to prove the existence of stem cells. If stem cells have specific marker genes, lineage tracing enables to label stem cells and their progenies in the same color using marker gene expression, using marker and LOXP recombination, which is very useful to show a long-term self-renewal and multipotency of the stem cells. Thus, discovering stem cell specific marker gene has been a crucial step to prove stem cell existence. To prove isthmus stem cells using lineage tracing technique, multiple groups tried to identify isthmus stem cell specific marker genes and reported 5 genes up to now. However, it turns out they are expressed not only in the isthmus region, but also in the base region, as shown in the example of ARIG1 lineage tracing experiment. Thus, there is no isthmus stem cell specific markers up to now. This promiscuous expression makes it very challenging to study isthmus stem cells. Would it be possible to prove existence of isthmus stem cells without specific markers, and how? To overcome the limitation I mentioned before, we thought, first of all, it was necessary to have a broad overview of clonal dynamics in corpus glands without bias to specific cell types and marker genes. To this end, we employed unbiased genetic labeling approach implemented by the Rosa confetti mouse crossed with Rosa Cree line. In this mouse, if tamoxifen is given, then all corpus cell or corpus gland cells can express one of the four colors, such as green, yellow, red, blue, in random fashion like this. The first, we observe the vertical expansion, vertical sections of stomatocorpus gland. Initially, the clones appeared almost everywhere in the gland, and the clones are segregated into large upper clones and smaller base clones gradually over time, up to 1.5 years. This shows that stomatocorpus are maintained by two independent stem cell compartments, as illustrated in these good examples. This is point number one of our key finding, because many people still believe one stem cell hypothesis for stomatocorpus gland. Then we also examine lateral expansion dynamics of clones. This is an example image of labeled gland in the isthmus region from the top. We also quantified the clone size by measuring the sector angle of clones. As a primer, I need to explain neutral drift model explaining stem cell dynamics in homeostasis for many tissues such as intestine, skin, germ lines. This is example of horizontal section of intestinal crypt. The one crypt is not maintained by just one stem cells. Instead, in each layer of crypt, multiple equipotent stem cells are arranged along crypt circumference. Here, different color means different clones. These stem cells compete each other until only one clone survives, leading to monoclonalization of individual crypt. As a result of neutral drift dynamics, in intestinal crypt, the number of surviving clones decreases, whereas the clone size increases over time, as you see here. This is exactly the same as what we observed from stomatocorpus, meaning a decrease of the number of surviving clones, but increase of clone size. This means that the loss of isthmus clone is completely compensated by the expansion of another isthmus clones, making the average number of labeled cells per gland remain constant throughout the whole lifetime. Thus, this implies the gastric corpus isthmus contains stem cells with long-term self-renewal. Furthermore, we paid attention to one more interesting point for stem cell dynamics in the isthmus region. The clonal dynamics in the isthmus region was much slower than that in intestinal crypt. We could still see many partially labeled clones in the isthmus region even after 3 months tracing. The quantification data also show the same point. However, we already know the vertical expansion in isthmus region is very fast, making us puzzled about the clonal dynamics in the isthmus region. To prove this paradox, we examine more carefully a cellular arrangement in the isthmus region. I'm going to show you one video file. In this video, the green cells are parietal cells secreting gastric axis. The red cells are Ki67 positive proliferating cells. As shown here, the proliferating Ki67 positive cells are intercalated between parietal cells. So thus we hypothesized these parietal cells may act as the barrier against the proliferating cells. To test hypothesis, we try to give damage to parietal cells by treating DMP777 to deplete them. We expected without this barrier, the clone dynamics is accelerated over time. As a result, we could see more number of fully lived glands at 3 months post-injury, validating our hypothesis. Based on this evidence and others, we suggest another model of stem cell dynamics, which we call as punctuated neutral drift dynamics. The punctuated model could reconcile fast vertical expansion and slow lateral expansion in the following way. Different from orthodox model, our punctuated model has a few barriers between different stem cell segments. If isthmus stem cells have fast cycling nature, the clonal expansion is faster vertically and laterally. That's why we saw a fast vertical expansion. However, different from vertical expansion, the lateral clone expansion is restricted by the presence of these parietal cells. Only when parietal cells happen to be lost, the clones begin to expand again and proceed for monoclonal state. This explains the slow lateral expansion. This is point number 2 of our key findings, because some people argue isthmus stem cells have slow cycling nature. To validate fast cycling isthmus stem cells include stem cells with self-renewal potential, we used the stasmin, one of the proliferation markers, and established stasmin-1 clear line crossed with Rosa-TD tomato to perform lineage tracing. As a result of lineage tracing, we saw clones appeared in the isthmus region and expanded fast in the upper region of a corpus gland. These clones survive long-term up to 6 months, showing a long-term self-renewal of stasmin-1 positive isthmus stem cells. By differentiation marker staining, we could show this clone contains multiple differentiation cell types, showing multipotency of stasmin-1 positive isthmus stem cells. We also characterized isthmus stem cells at molecular level using a single-cell transcriptome analysis. To this end, we isolated stasmin-1 EGFP positive cells from stomach corpus tissues and generated single-cell RNA-seq data using a plate-based method. First of all, the cells are segregated into 2 groups, stasmin high cells, high population, and stasmin low population. These stasmin high cells are segregated again along cell cycle phases, whereas the stasmin-1 low cells into differentiation cell types such as P cells and NEC cells. The pseudo-time analysis suggests that a bifurcation of stasmin high cells into PIT and NEC cell lineages in stasmin-1 low cells. Furthermore, we also observed proliferative heterogeneity in stasmin-1 high cells in terms of differentiation marker gene expressions. So when we further dissected stasmin-1 high cells, after cell cycle regression, stasmin-1 high cells are grouped into 6 subclusters. The pseudo-time analysis showed that subcluster 2, 3 are biased to P-cell lineage, subcluster 6 biased to next cell lineage. However, cluster 1, 4, 5 are unbiased to neither of two sublineage. This analysis suggests that subcluster 1, 4, 5 are likely to be stem cells, and subcluster 2, 3, 6 are sublineage restricted progenitors of stem cells. Then we try to find a gene expression specific to the unbiased subclusters along these two pseudo-time paths to identify isthmus stem cell specific markers. We saw these two major points. Point number 1: there are enriched but not exclusive expression signature of isthmus stem cells, such as the PSMA7 and DFB8 and CIB1. In other words, different from our initial expectations, we couldn't find any single marker gene specific to isthmus stem cells. Point number 2: unbiased clusters expressed two sublineage markers, MUC5AC, MUC6 at a medium level, and begin to turn on one gene and turn off the other gene along the sublineage specifications. Our RNA-scope result in the isthmus region confirmed this second point. In summary, the corpus gland of adult mouse gastric epithelium consists of two stem cell compartments, one in the upper and the other in the gland base. Especially isthmus stem cells follows punctuated neutral drift dynamics in homeostasis. At molecular level, isthmus stem cell was not defined by single specific marker gene expressions, rather by a co-expression of multiple marker gene expressions. This new model could be applicable to other examples showing very slow monoclonalization of as in the case of human colon. This also emphasized the importance of physical environment in stem cell fate behaviors rather than intrinsic program of stem cells. This project was carried by the close collaboration between Professor Ben Simon's lab and Dr. Bongyong-Koo lab. I'd like to thank members of two groups who helped this project, and also to these funding bodies that supported this project. Thank you very much for your attention.